So how is genetic testing used in HIV care and research? Well, this video helps explain how genetic science is currently used, how it might be used in the future, and also its limitations. The human immunodeficiency virus and acquired immunodeficiency syndrome still remain one of the biggest global challenges. However, we now have highly effective treatments for HIV, which have completely transformed the outcomes of having this disease. This is a pretty remarkable achievement, and is a result of dedicated work from scientists around the world. Much of the research in HIV is focused on its genetics, which has revolutionised our understanding of the virus. However, this is a complex subject with complex terminology, so it's useful to go through the basics of how this science actually works. HIV, like you or I, is formed by and contains long lengths of genetic material known as nucleic acids. They are formed from units called nucleotides, which are essentially the building blocks of all genetic material. In humans, this material comes in the form of DNA. In HIV, it comes in the form of what's known as RNA. As with humans, the genetic material within individual viruses is unique, but is most similar within its own viral family. This fact allows us to work out how different strains of viruses are related and where they might have originated from. Two terms are important here: phylogenetics and genetic sequencing. Phylogenetics is a biological technique which determines evolutionary relationships between different organisms based on shared common features. Genetic sequencing is the process of finding out that unique order or sequence of nucleotides that make up a length of DNA or RNA. So let's look at phylogenetics. In HIV phylogenetics, the genetic sequences of different viruses are compared to one another in order to determine how closely related they are. With the addition of complex mathematical algorithms, a phylogenetic tree is produced, which shows an estimation of the evolutionary history between different viral strains. Like a family tree of, say, the British royal family shows the relationship between Queen Elizabeth and Queen Victoria, phylogenetic trees likewise show the evolutionary relationship between different viruses. And so closely related groups of viruses might mean recent transmission between them. Phylogenetic analysis can be used to look at patterns of transmission of the virus within a certain population to see where, when, and how HIV is spreading. This can really help guide HIV prevention strategies. With improving techniques, it can now be used to detect and analyse new outbreaks of HIV in real time, which should allow us to better control and slow the spread of the virus. The data used in phylogenetic analyses comes directly from the viruses in question, and this is where genetic sequencing comes in. Within the UK, the majority of our HIV genetic sequence data is a result of routine drug resistance testing in those who are HIV positive. A section of the viral genetic sequence, taken from a blood sample, is isolated, then amplified to generate lots of copies. The section of data that's taken is the area of the HIV gene where we know the majority of mutations that can cause resistance to antiretroviral drugs are present. This technique is useful as it's cheap and widely available, but the downside is that only one section of the entire sequence is analysed. Therefore, if we only sequence one part of the genome, this means a lot of data isn't analysed. It's a bit like reading a book that has pages missing. It provides some information, but not the whole story. Therefore, new techniques called next-generation sequencing have been developed to allow us to get more information from genetic data. The two most important techniques are whole genome sequencing and deep sequencing. The former, as the name suggests, is a technique which allows scientists to sequence the entire genome of a virus, therefore allowing us to read the whole book, not just a small section of it. Deep sequencing is a technique that is able to detect mutations that occur at very low levels that might not necessarily be picked up using standard techniques. Both of these have improved the amount and quality of genetic data we can obtain from viral samples. You may have heard about the relatively recent use of phylogenetics in legal settings, where an individual has been accused of deliberately or recklessly infecting another person. Although phylogenetics has been used, it can't definitively prove direct transmission of the same virus from, say, person A to person B. So the benefit of using it in this way is still being debated. However, a distinction that must be made really clear is that the use of sequencing and phylogenetics in legal cases is completely separate from how they are used in clinical care and research. The genetic data taken from patients with HIV in clinic will only ever be used to help with their care and anonymously in research projects. It is never used to link the spread of the virus to individuals. So if you've had a HIV genetic test in the past, or are having one as part of your current care, hopefully this video has helped explain how the data is used and will reassure you that results are, and always will be, completely confidential. We hope this video was useful, and if you have time, please fill out the survey in the link to give us some feedback. Thanks for watching.